Hello, I am Marco Ruggiero, although this voice is borrowed from AI for clarity of exposition. Today, I want to share the results of a scientific study that has just been published. Scientific research has taken a decisive step in understanding neurodegenerative diseases with the publication, just a few days ago on April 20th, 2026, in the prestigious international journal Nature Medicine, of a study that rewrites the paradigm of early diagnosis for Parkinson's disease. This work, titled Microbiome Signature of Parkinson's Disease in Healthy and Genetically At-Risk Individuals, represents a milestone for the scientific community. It demonstrates with analytical rigor that our gut speaks long before the brain manifests signs of decline. As a researcher who has dedicated decades to the study of the molecular biology of the microbiome, I observe with extreme interest how the microbiota gut-brain axis has evolved from a suggestive hypothesis into a fundamental pillar of modern medicine. Parkinson's disease is currently the fastest-growing neurodegenerative pathology globally, with an economic and social burden that threatens to become unsustainable in the coming decades. One of the greatest obstacles in the fight against this disease has always been late diagnosis. By the time classic motor symptoms like tremors or rigidity appear, over 50% of dopaminergic neurons have already been lost. However, this new study tells us that alterations in the gut microbiome can identify individuals at risk long before the manifest phase, acting as a true biological sentinel. The team of researchers from institutes worldwide examined a large international cohort, analyzing not only diagnosed patients but also healthy individuals and carriers of variants in the GBA1 gene. This gene is the most common genetic risk factor for Parkinson's disease, yet only about 20% of carriers actually develop it. The question science could not answer was why some fall ill and others do not. The results published in Nature Medicine suggest the answer lies precisely in the balance of the bacterial flora. It was discovered that gene carriers who do not yet manifest symptoms have a microbiome in an intermediate state between healthy subjects and Parkinson's patients. This means the microbiome evolves alongside the disease's progression, becoming a prodromal marker of inestimable value. But what exactly changes in our gut? The research highlighted a significant loss of bacterial species belonging to the Lachnospiraceae and Ruminococcaceae families in both at-risk subjects and patients. These bacteria are not simple guests. They are true producers of butyrate, an essential anti-inflammatory substance for protecting the nervous system and maintaining the integrity of the intestinal barrier. When these protectors decrease, it opens the way for a state of chronic inflammation that can favor the accumulation of toxic proteins, such as alpha-synuclein, which then migrate from the gut to the brain. In parallel, the study noted an increase in bacterial species associated with inflammatory states, such as Streptococcus mutans and Ruminococcus ganavos. A revolutionary aspect of this work is the identification of a specific score, named PDMS-16, based on the presence or absence of 16 key bacterial species. This score proved capable of accurately distinguishing individuals who, while clinically healthy, already show a biological profile and invisible symptoms, such as sleep disorders, depression, or chronic constipation, that precede Parkinson's. Furthermore, the validity of these results was confirmed by analyzing independent cohorts in the United States, Korea, and Turkey, demonstrating that this microbial signature is a global phenomenon. The message for researchers and the public is clear. Microbiome diversity and richness are not just indicators of good digestion, they represent the cornerstone for neurodegenerative prevention. Maintaining a varied and healthy intestinal ecosystem through a quality diet and targeted bacterial recolonization. Interventions could slow down or even stop the degenerative process before it becomes irreversible. It is interesting to note that the study shows a direct correlation between diet quality and microbiome health. Those who consume more fruits and vegetables show less severe microbial alterations, confirming that lifestyle is a fundamental ally of genetics. In conclusion, this very recent work offers us a new perspective. We must no longer consider Parkinson's or other forms of dementia exclusively as brain diseases that suddenly appear in old age. They are the result of a systemic imbalance that begins in the gut years, if not decades, before tremors or memory loss. Protecting our microbiome's biodiversity today means protecting the integrity of our cognitive future. As a researcher, I see in these data confirmation that the medicine of the future will increasingly be a medicine of prevention based on the meticulous care of the microbiome, our internal bioreactor.